about CRISPR-Cas, where it comes from, where it stands, and where it's going. The gene editing tool CRISPR-Cas comes from bacterial immune systems. Scientists were just doing basic research trying to study these bacterial systems, and they discovered that you could actually program these specific proteins with RNA guides that would take them to DNA targets and then cut the targets, and then you could use that as a tool to introduce specific changes you wanted to make. The tool has since evolved so that we can make more specific changes, but it still has its challenges. And so we're going to talk about some of those challenges, about where the technology stands right now, and about some of the things that are coming in the future. I'm going to start by talking about CRISPR-Cas as a gene editing tool. And then later I will go back and talk about it as a bacterial tool, but I figure more people care about it as a gene editing tool. And so I will start with that stuff. And I think it's important that people kind of know what it is, know some of its limitations, and know the new stuff that's in the works. And so just the basic bottom line, it comes from this bacterial immune system. And these bacteria, they're like, okay, so this virus infected me. I want to not let that happen again. So I'm going to take some of that viral DNA. I'm going to integrate that into my genome. So I'm going to stick a piece of that viral genome into my genome. And then if the virus comes to attack me, what I'm going to do is I'm going to use that sequence that came from that virus in order to recognize the virus. And the way that I'm going to do this is I'm going to have this Cas protein. And this Cas protein has scissors, so it's a nuclease. It'll cut the DNA of the virus that infects it. And how it knows where to cut is it's going to bind onto this CRISPR guide RNA that's going to contain the sequence from that virus. What we could do in the lab is we could take a Cas protein and then we could give it a guide RNA. But instead of targeting a virus, we're going to have it target whatever sequence of DNA we want to cut. And so we give it these specific CRISPR guide RNAs with bind to this Cas protein. The Cas protein goes to the site and cuts. Well, at least the traditional Cas protein does that. So like the original Cas system that was developed as a laboratory tool was Cas9. And what it does is it goes to the target and then it cuts both strands and lets the cell take over. So when you have one of these, like both strands of the DNA are cut, so this is a double-stranded break. What's going to happen is that the cell is then going to kind of freak out. If you don't give it any alternative, what's going to happen is you typically get a knockout. So the cell's going to do something called non-homologous end joining. Basically, it's just going to try to stitch those ends together. And this typically leads to what we call indels, so insertions and deletions. And this typically inactivates the gene. And so what we say is we get a genetic knockout. So functional protein is not made from this place anymore. A lot of times, however, you want something more than just knockout. You want to actually just change the sequence that's there, maybe fix a typo in that genetic sequence. A way that you could do this is if you give it a new template to put into place. What you can do is you can use what's called homology-directed repair. Here you provide an alternative template that has the ends of that template match the ends of kind of like the cut sites. And that way, through homology-directed repair, the cells will be like, hey, that looks like that, and that looks like that, and let me put those together. And voila, you get homology-directed repair. So that can work sometimes. The problem is it's limited to certain cell types. So basically that machinery is only really active during cells that are actively dividing. It's also not very efficient, and making those double-stranded cuts in the DNA can kind of make the cell freak out. And it can also lead to changes you don't want, like pieces of chromosomes getting swapped, and lots of things that could potentially even do have outcomes like causing cancer if they're in the wrong place. And so the technology has kind of been moving away from this more conventional CRISPR-Cas. So in the lab, it's great a lot of times, but if we want to use it for, say, therapeutics, we want something that's more precise, something that's more efficient, and something that is, we can make a wider range of edits, say. So introduce nickase Cas. So when we say a nickase, that's saying it's that one of the pairs of scissors is inactivated, so it only cuts one strand of the DNA. So Cas has basically two pairs of scissors, the original Cas, the wild type, the non-modified. So it cuts double-stranded DNA. It cuts both strands. And so it has two different domains or basically parts of the protein. One of them cuts one strand and the other cuts the other. You can inactivate either of those pairs to get different forms of the protein that will either cut the strand that the guide binds to or cut the opposite strand. And then what you can do is you can get a single cut. When it comes to these NCAS or nickase Cas proteins, they're typically attached or conjugated to another protein, an enzyme that's going to actually do something to modify the DNA. Because, like, if you make one strand, the cell's just like, okay, I'll just stitch it back up, no big deal. But if you have something that modifies the DNA, when you cut it, then you can kind of have a way to introduce changes that you want. And so there are two main forms of this that are typically used. There's base editing and prime editing. Base editing, what it does is it uses nucleotide deaminases to basically alter single nucleotides to make point changes. And so what this allows you to do is say in the case of an adenine base editor, you would deaminate the adenine. That would give you hypoxanthine, which is the base for inosine that gets read as guanine. If you have cytosine and then you deaminate it, you get uracil, and then that uracil is going to be read basically as thymine when uracil is in RNA, but the cell will put in a thymine. And then the opposite strand, you're going to get an adenine. And so you get both strands changed. And if you want to learn more about them, I recommend this AdGene blog. There are also newer versions where you can also do other changes. So in addition to transitions, you can do transversions. And so I will point you to resources to learn more about that. You might be wondering, why not just use a catalytically dead Cas protein for this? Why do you actually need to sniff the DNA? A couple of reasons. One, with the catalytically dead Cas proteins, there's less specificity because the requirements for actually cutting are going to be stricter than the requirements for binding, which is why you can get more off-target effects with the dead Cas proteins. Also, by making that nick, you're kind of differentiating the two strands. And so you have the nick strand that the cell is going to be like, oh, that's the damaged strand. So I'm going to use the strand that was just edited as a template for fixing that other strand. So base editors are great if you just want to make a single change. So if you're trying to correct like a point mutation, maybe the patient only has a single nucleotide that's wrong in their gene, voila, let's go in and fix it. But if you want a bigger change, say you want to make a large insertion or deletion, well, here you want to use something else. This new technique called prime editors, they use these longer guide RNAs called PEG RNAs, and that's the prime editing guide RNA. What it does is it basically has a template, and then the Cas protein is attached to a reverse transcriptase. And so this guide RNA has a template for the sequence that you want to put in. So rather than give the insertion piece as a separate piece of DNA, you're going to give it as part of this RNA guide. Now when the guide binds the Cas protein and the Cas protein binds to the DNA, the reverse transcriptase part is going to be used to make a DNA copy from that RNA guide, and then that's going to get inserted into the sequence. So a little more complicated, but you are able to then get this repair and you have the insertion or you have the deletion or whatever change you want to make, you have incorporated. And so those are kind of the basic strategies with these nickases is that you have some sort of modifying enzyme on a Cas protein that is a nickase. It still needs a guide RNA to direct it to where you want it to go. And then the Cas protein will take over with whatever is associated to that Cas protein. There are also tools being developed to make even larger insertions, so transposons associated with CRISPR and integrases. I'm not going to go into the details, but I'll provide you to resources for more. You also see this like DNA polymerase editor. So rather than a reverse transcriptase, this one actually uses a DNA polymerase. And that avoids some of the problems for, say, when you have that RNA template, because RNA can form more interactions, it can have unwanted folding. And so the DNA is gonna help with that as well as some other things. And so that's kind of one of these up and coming technologies that you might hear more about in the future. So For the genome editing strategies, our basic strategies, we have the conventional, we're the Cas. So when we say Cas, there's different Cases, and we'll talk more about different Cases. Cas9 is that kind of traditional one, that original one, that one that goes and it makes the two cuts. Then the cells take over. If you don't give an alternative, you just get the ends stitched together through non-homologous end joining. That typically causes these like uncontrolled insertions and deletions, so you get gene inactivation or knockout typically, but you have to go and check that you actually made the change that you want. And so typically people will do things like Western blots in order to verify that the protein is no longer being made. If you give it an alternative, you can have homology-directed repair. And here you're going to give it an alternative template. The ends of that template match the cut site region. Problem with this is it's inefficient and it's limited mainly to actively replicating cells. This kind of second generation of CRISPR-Cas genome editing systems, these use a nickase Cas, so an NCAS. It makes a single-stranded DNA break and is attached to a DNA modifying enzyme. With base editing, you're going to change a single nucleotide base, and it's good for point mutations. With prime editing, you're going to stick in a newer alternate sequence that's provided as part of the extended guide RNA, so that PEG RNA. And you can do that because it uses an attached reverse transcriptase to make a DNA copy of that template. So that's great. We have a lot of genome editing strategies. I will talk more about some of the limitations to these strategies in addition to the ones I mentioned before. But I also want to say that CRISPR isn't just for genome editing anymore. So the strategies that we talked about are all for like genome editing. So they were editing the actual DNA, which means that the cells that are made from that cell will be modified. Now, if you're just editing cells in your body, other than your germ cells, so not your sperm, not your eggs, the cells that come from those cells, so when that cell like replicates, the cells that are derived from it will also have that modification. And that cell will have that modification for the rest of its life. However, like the next generation, those changes won't get passed on. If you do germline editing, then those changes would get passed on, and that's what's more kind of ethically dubious. The somatic cell, so just like the non-germ cell editing, it's good in that it's not going to get passed on if you're worried about it getting passed on. But the problem is that you have to target all of the cells that you want to be targeted. And so if you target, say, the stem cells, then that's going to allow you to have that change more widely. But sometimes you want to target just a single tissue, and so we'll go more into targeting in a little bit. But sometimes you don't want that permanent change, and so you want to make something more temporary, either for safety concerns or just because you don't need a permanent change. And so there are tools that you can basically use a catalytically inactivated or dead Cas. And so you see this referred to as dCas. So dCas, it doesn't cut, it's catalytically dead. And so it doesn't have those scissors, it can't do any cutting, but it still has the ability to bind the guide RNA, which then has the ability to take it to the target DNA. You can then, if you have that Cas protein, that dead Cas protein attached to something, you can go ahead and you can make these temporary changes. Some of the changes that can happen are to allow for transcriptional regulation via transcription factors or epigenetic changes through chromatin remodelers. So we talk about epigenetic, epi over, above, and so this is changes to like modifications to the DNA and the way the DNA is wound that allows for differences in the expression of the gene without changing the actual underlying nucleotide sequence. Some of the acronyms associated with this sort of temporary changes are CRISPR-I, CRISPR inactivation, CRISPR-A, CRISPR activation. These are going to target transcription factors to the site. There's CRISPR-on and CRISPR-off. These modify the chromatin. So let me talk really quickly about this. So you have a lot of DNA, like a lot of DNA. In order for it to fit in your cells, it has to be tightly wound around little proteins called histones. And this is what we call chromatin when we have this DNA wound around these histone proteins. In order to actually get into the DNA to transcribe it, so to make RNA copies of it that can then be used for translation to make protein from it, you need to actually get access, which means you need to unwind those little coils. And the way that it happens is that you have chromatin remodelers that can go and actually move those nucleosomes, move the DNA wound around the histones, kind of move it out of the way so that the transcription machinery can get in there. And so you can have chromatin remodelers that actually go and change the proteins, the histone proteins. You can do things like methylation, and the methylation often is associated with silencing of a gene. And then the acetylation, which basically is associated with activation. It's not quite so simple, but when you activate it, you are basically neutralizing the histone protein's positive charge, and that positive charge was helping it stay tightly wound to the negatively charged DNA. And so the modifications can then make the DNA open up. Once the DNA is opened up, then you still often need transcription factors, so proteins that come and help recruit the machinery to do the transcription. And so transcription factors are these proteins that come and help the RNA polymerase bind where it's supposed to bind or prevent it from binding where it needs to bind. And then the chromatin remodelers will actually kind of give it space. And so both of these can be used as strategies to alter the gene expression. And that's altering the expression, that's not altering the actual DNA sequence itself. And so the epigenetic changes, they can be passed on a little more, but when you're just having things like transcription factors, that is more of a temporary cell-restricted thing. In addition to changing the gene expression, changing the sequences of the DNA, there are also different Cas versions, so like Cas13, Cas711, that could be used to target RNA for modification and/or inhibition. And you can actually go and make RNA changes. And I'm not going to talk about that, but I will direct you to resources for more on that. And then there are also techniques that we can use to use CRISPR-Cas for diagnostics. So I had talked a little about this during the times when things were really bad with COVID and people were trying to find quick, easy ways to test for the presence of the virus. Basically, you can use Cas proteins that get activated to cleave reporter RNAs by binding to targeted sequences. So you can use reverse transcription to make DNA from the RNA, make copies of the DNA, and then the Cas protein will go and it'll bind to that DNA copy of that viral RNA and cleave it. And this will then, once it cleaves, it kind of gets the Cas protein in this activated state that it can go and cleave a labeled reporter. This can then be used to test for the presence of a virus. It can also be used to test for the presence of other nucleotide sequences, and so CRISPR-Cas diagnostics are kind of a cool tool that you can use CRISPR for. That's not what you conventionally hear about it used for. So let's talk more about what you probably are more used to hearing about, and so that's CRISPR used for gene editing to cure diseases. And so the other day I did a video about the world's first patient treated with personalized CRISPR gene editing therapy. And basically, this baby had this metabolic disorder called CPS1 deficiency. They had this problem with this enzyme in the urea cycle. And the urea cycle is used by your body to remove of amino groups safely without having the buildup of ammonia. And so when you don't have this protein, you build up ammonia, that damages the brain, bad stuff. The doctors were actually able to go and correct this error in the DNA of this baby that was causing this disease using base editing. And this was a breakthrough because it was developed specifically for that baby's particular mutation. So it's a really rare disease, and they were able to develop this treatment designed specifically for that baby. You might have also heard about a non-patient specific, more generally applicable, but still not totally CRISPR treatment for sickle cell disease and beta thalassemia. These are disorders of hemoglobin, which is the protein that transports oxygen through the bloodstream. And so when people have mutations in this protein, they can't transport oxygen efficiently. And in the case of sickle cell anemia, there's a mutation in the hemoglobin protein that actually causes it to stick together. And that causes the cells to get misshapen into sickle shapes. And those then get caught in the blood vessels and cut off the blood flow. And so that leads to painful sickle cell crises. Now, your body actually has multiple forms of hemoglobin. And so there's a hemoglobin, there's a fetal form and an adult form. And so the fetal form has different properties than the adult form that make it better for allowing the fetus to grab as much oxygen as it can from the blood the mom's already taken the oxygen from the blood. And so the fetal form has these different properties. And so it gets shut off after birth when the adult form kicks in. However, all the cells still have the instructions for making that fetal one. And so what scientists can do is they can actually get the fetal form expressed. And the way that they do this is often in this one treatment, this approved treatment. this Casgevy is they prevent an inhibitor from binding to that sequence for the fetal hemoglobin that allows the fetal hemoglobin to be made. And even in these adults, and then this restores the functioning of the hemoglobin. That's an example of ex vivo treatment where you're removing the cells from the patient, altering them, and then putting them back in. You're able to do this with blood disorders because you can take out bone marrow, fix the bone marrow, put the bone marrow back in. The bone marrow is where the blood cells come from, and so now the blood cells that get made will have the alterations. You can also do this with strategies for treating, say, cancer. You take cells from the patient, like immune cells from the patient, and you alter them so that they target the cancer. And then you put them back in the patient, and then those cells, those immune cells, will go and fight the cancer. So you might have heard of like CAR T therapy, that sort of thing. In the case of the baby with the CPS1 deficiency, that was actually happening in vitro. And so they were actually going and treating the baby by giving it the CRISPR-Cas machinery that then targeted the liver and allowed for the changes to be made inside of the baby without having to take out any cells, without having to do any bone marrow transplant, all this stuff. And so the liver is commonly a site that's kind of like more easy to target. And so with like LNP, so lipid nanoparticles, your liver has a lot of receptors for lipoproteins and things. And so it's easier for these molecules to get to your liver. And your liver is also kind of like where things go as a first stop. It's your detox center and things. And so it's easier to target things to the liver. You can also target things to like the eye. So some CRISPR-Cas treatments have been used for like the eye because you can basically just inject things in there and it's got barriers that are going to prevent it from making too widespread of changes in other cells. But other tissues are harder to target, and so targeting is one of the things that is one of those problems with CRISPR that needs to be addressed. And so people are looking into things like, can we add little proteins on the outside of these nanoparticles that then will direct it to specific tissues? So maybe they do antibodies or things like this. But in the case of this baby with the CPS1 deficiency, because the problem was in the liver, they were able to give it these lipid nanoparticles that contained the mRNA for making the Cas protein. I'll talk more about this, but typically the RNA is delivered rather than the protein, allowing the cells to make more of the protein than you could if you were just trying to stuff in the protein. And the RNA can be more stable because you could do modifications and things. And it's easier to sneak in. It also has the guide RNA and then the lipid nanoparticle. More about delivery in a bit. All of that was kind of in vivo or ex vivo as opposed to in vitro. And so in vitro is not in a living organism. Instead, it's in a test tube or cells. So though some people will call like cells are kind of like an intermediate zone. And so for bacteria, a cell is in vivo, but sometimes people refer to like in cellula or in culture. The basic idea is, though, it's not in a living organism. And so what you could do is you could actually use CRISPR screening, either permanent like knockout or with the CRISPR-I or CRISPR-A, where you're inactivating or activating the gene. And that's good if you're wanting to test for effects on essential genes. So if you knocked it out, the cells would just die. But what happens if you inactivate it? Or you inactivate it at a specific time? So in CRISPR screening, what you do is you have a big library, so a big collection of RNAs, and you infect cells with them, or you get those into cells somehow. Each cell gets a different one of these guide RNAs. You then do some sort of tests, like maybe you're seeing if they're surviving, maybe if they can resist a drug, maybe just trying to see what effects it has. And then you go and you look at which of the cells have a trait that you're looking for or don't have a trait that you're looking for. And then you can go and sequence the guide RNAs from those cells in order to see where which guide it was that caused the thing. CRISPR can be used for both reverse and forward genetics. So with reverse genetics, we change the gene and observe the effects of the phenotype. With forward genetics, we observe a phenotype and then find the responsible gene. And so in this case, we're basically using forward genetics because we're making the changes to the gene, but not in a specific way. Instead, we're observing some phenomenon that comes from a change that we made and then going to find what that change actually was. We can also use reverse genetic techniques where we know what change we want to make, and then we want to see what effects it has. This can be great for making things like mouse models or even tissue models. All of these things that are more realistic than just working purely in vitro. What you can do is you can use CRISPR-Cas editing in order to knock out, so basically remove specific genes. So when we say knockout, that's we're permanently inactivating the gene, so you're not making the protein anymore. Or we can knock in where we stick a gene in its place. And so both of these can be used to do things like make mouse models, make cellular models. So you can take, say, skin cells from a patient and you can convert them into what we call induced pluripotent stem cells, so iPS cells. And those can then be differentiate. You can get them to differentiate, change, mature into different cell types. And you can actually, though, make changes to the DNA before you do the differentiation, and then you'll have patient-derived cells that have a change that you would like to see. And this can then allow you to get different cell types than you would normally be able to get. And that would be just from a skin cell, and you can make those changes in the cells of the actual patient based just on those skin cells and study the effects that, say, these changes would have in different tissues, perhaps. So that's kind of like one of the things that you could do. But CRISPR is not without its limitations. So let's talk about some of the challenges. The main challenges, so I really like this article and it's open access, and I will provide a link, but it goes really well into some of the challenges and solutions. And I want to talk more in depth about each of these. The kind of four main ones they point out are specificity, targeting scope, controlling editing outcomes, and deliveries. I kind of summarize it this way. We want our editing to be accurate and efficient. And so accuracy, we want to prevent off-target activity, so unwanted edits. And we want to be able to target any sequence that we want. I'll talk more about this, but there's this thing called PAM that's going to limit this. We also want to be able to expand the range of edits we can do. So we want to do single base changes versus large insertions. We want to be efficient, so we want to target all the cells basically, or at least all the cells that we want to target, which gets back to targeting specific cell types. We want to be able to deliver that machinery, and we need to do so without triggering the immune system. So those are some of the challenges, and we'll talk more about these and more about their solutions. Part of the solutions comes from remembering that CRISPR-Cas comes from a bacterial immune system. The CRISPR-Cas system originally used in the lab and still conventionally used, that original kind of Cas9 system, comes from Streptococcus pyogenes. But other bacteria also have CRISPR-Cas systems. And so if we want to make different changes, we can do things like turn to different CRISPR-Cas systems and look to different Cas proteins. We can mine the bacterial genomes to find naturally occurring Cas proteins, or we can alter existing Cas proteins. This can be directed through targeted mutations, so maybe look at the sequence, look at the structure, try to predict what mutations would make changes that you want, and/or directed evolution. So basically, you do sort of random mutagenesis in the lab while looking for a desired outcome. The changes that are already being used are doing things like using nickase Cas proteins instead of the fully active ones to minimize off-target damage and cellular responses. You can also do things like modify the guide RNAs to decrease off-target binding to avoid that off-target effects, and improve the delivery systems, such as using different LNP formulations, so those lipid nanoparticles, or using cell-specific targeting factors. So let's go into all of these. Starting with specificity. It's really important that the CRISPR-Cas editing is specific. We don't want that off-target editing activity. That can be catastrophic. It can cause cancer, and it can cause other problems. And so testing for off-target activity is a huge, huge, huge deal. And so scientists often have to screen a lot of different guides in order to find the best ones to use. So going back to that baby with the CPS1 deficiency that they made that special treatment for, this is from their supplemental of their paper, and you can see that they use this tiling approach to test guide RNA spanning the region of the mutation that in the baby's gene that then they wanted to correct. And then they went and tested them all. What they were testing for was like efficiency, so how well it was actually making the edits, how efficiently it was making the edits, so like what proportion of the cells actually got the edit you want, as well as off-target activity, so was it making changes elsewhere. They used a variety of methods. They prioritized sites that were likely to be to be off targets based on matching similar sequences. And then they actually went and generated cell lines and did all this testing in order to be able to show how specific the treatment was. So lots of work goes into making sure you don't have off-target activity because it can be so catastrophic. One challenge is that not all sequences can be targeted easily. And so this gets to the targeting scope, or this thing called the PAM, that has to do with this thing called the PAM, the protospacer adjacent motif. I'll get more into this when I talk about the bacterial origins, but the protospacer is the part of the target DNA that matches the guide RNA. And more specifically, it matches the spacer part of the guide RNA, which is the unique part of the guide RNA. Cas9 is a pretty boring protein until it binds to that guide RNA. Then that kind of snaps the protein into a tension. Cas then goes and it kind of hops along looking for that PAM. If it finds that PAM, that protospacer adjacent motif, which is a part of the target DNA that matches the Cas protein, and when I say matches, like the Cas protein is a protein, it's not nucleic acid, so it's not going to have just like simple base pairing complementarity, but it can have favorable interactions with the protein. What's going to happen then is if the Cas finds a matching PAM, it'll unwind the DNA, and then it'll cut the DNA. The PAM sequence is specific for the Cas protein. In the case of this Streptococcus pyogenes Cas9, it's going to be NGG. So where N is any nucleotide, so an N and then two Gs. So it's a pretty generic sequence. It's found fairly frequently throughout the genome, but if you're in like an AT-rich region, then you're not going to have a PAM nearby. And you need to have that PAM nearby in order for the edits to be made. So people have turned to different Cas proteins that have different PAM sequences. So remember, the PAM sequence is making favorable interactions with the Cas protein. So if you change the Cas protein, you'll change what sequence it likes in terms of its PAM. You can also make changes to the Cas protein itself to change its specificity. More often, these kind of like relax the specificity, but relaxing the specificity then increases the risk of off-target activity, and so that could be a problem. But you can see here, like this Cas12a, it uses a different PAM. And then you have different natural versions of Cas, as well as then different mutated versions of the Cas9. When you see these kind of abbreviations in front of Cas9, that's talking about versions that come from different species of bacteria. And so Cas9 is the protein, and then this is telling you kind of what species of bacteria it was coming from. The edits need to be controlled, predictable, and efficient. When you make those double-stranded breaks, you're basically just saying to the cell, okay, take over now, and trusting that the cell can do it well. There's problems with this. One is that if you don't provide an alternative, you're just allowing it to stitch things together. It's unpredictable how much it's going to insert, how much it's going to delete, all these things, because it's going to go and it'll kind of alter the ends to try to do its best to fix things, but it can't really do it all well. And so you're going to end up with kind of unpredictable changes. If you give an alternative, you can have that homologous replacement, but that's not very efficient. It's restricted to certain cell types, and it can also introduce unwanted mutations during the process once doing that fixing. Solutions to try to kind of make it more efficient are to inhibit the non-homologous end joining. So inhibit it from just stitching together things and trying to encourage it to do the homologous recombinations, such as tethered repair templates, doing it in controlling the cell cycle so that it is you put it in the phase at which the cell is actively dividing, that sort of thing, as well as turning to these nicases that are able to just cut a single strand and then make these specific changes. Because you're only cutting a single change, you're less likely to have these uncontrolled outcomes. But you still have to worry about the efficiency of the outcomes that you do want. So is it making the changes that you do want? Not only is it not making changes that you don't want. Because if you stick something in that doesn't do anything, well, you're not going to have any changes that you don't want, but you're also not going to have any changes that you do want. In order to have any changes, you have to actually get this machinery into cells, and that could be a challenge. The machinery is typically delivered as RNA, and so that the cells are basically going to have to make the Cas protein itself. And so you give the cells the messenger RNA for making the Cas protein, as well as the guide RNA, And then you have some sort of encapsulating method that allows it to get into the cells. So this is again coming from that CPS1 CRISPR treatment paper. And I'll direct you to the video I did on that if you want to learn more. But you can see that they have this mRNA that is making the Cas protein. They have this Cas-nickase gene next to this adenosine deaminase domain. You've got your cap, your tail, your UTRs, all this great stuff. There's also these modifications, and so all the uridines are modified to methyl pseudouridine. And I wanted to point this out because Katalin Karikó, who won the Nobel Prize for her work, part of what she won the Nobel Prize for was determining large part of what she won the Nobel Prize for was determining that sort of unmodified RNAs are immunogenic, and that if you modify the uridines to pseudouridines, then that prevents the cells from freaking out when you give it to them. And that paved the way for mRNA vaccines, as well as for things like this, because now we're able to make these modifications to the messenger RNA for making the Cas protein. Then the cells can make the Cas protein, and then they can go and they can do their thing. There are challenges with this. One is that the mRNA, even though like the cells can make a lot more protein from that mRNA, so on the whole, you get a lot more out of it than if you were to put in the protein. RNA is big. And not only do you have to stick in the Cas9 RNA, but you also have to stick in the guide RNA. And sometimes what you want to do is you want to actually integrate the machinery for making the Cas protein in the cells, like when you're doing things in the lab. When you have those long sequences, that can be a challenge. And so some of the optimization has been trying to find Cas proteins that are actually smaller. They have shorter mRNAs, they have shorter DNA genes that can then be delivered more efficiently. With any of these, however, you still need to be able to get the genetic information into the cells. And so when we talk about getting genetic info into cells, we call that transfection. If you work in a lab, you probably do things when you're sticking like DNAs, maybe you're sticking plasmids into bacteria cells. We often refer to that as transformation. And so transformation, however, we don't want to use that term when we talk about human cells and things because it has different connotations. And so we typically just use this word transfection. In the lab, you might be doing things when you're working with bacterial cells, you might be doing harsh things like heat shock with chemically competent cells. Laboratory methods for delivering genetic information into mammalian cells and things often uses electroporation, where you're using electric charge to get the charged DNA into the cell. Both of those are kind of more laboratory techniques. In the body, we typically use things like lipid nanoparticles or viral delivery methods. Often these formulations for these lipid nanoparticles have these cationic lipids that are actually ionizable lipids that are actually going to, when they get inside of the cells, they get swallowed into these endosomes, and then the pH changes, and then that allows the lipids to kind of change their interactions and allows the RNA to get out. So really cool formulations, and that's another room that there's advances in all the time. There are also viral delivery methods, and so viruses are really good at getting genetic information into cells, and so we could take advantage of that in order to stick information that we want into a viral kind of coding and then stick that into cells. There are problems, however, because the body might recognize the virus as a virus and attack the virus and not allow the mRNA to actually make the Cas protein and make all that stuff. There's also problems even if the body doesn't recognize it as a virus the first time. If you try to give another dose, well, then it's going to recognize it. And so more of these lipid nanoparticle strategies, things like that, is typically the way that things are going. But you might see things that use, say, adeno-associated viruses or adenoviruses. Some of it depends on what kind of cell types you're trying to target. There's all sorts of things. And so it's complicated, as most things are when you're talking about biochemistry, when you're talking about things, especially in biological systems. But bottom line, CRISPR-Cas is a powerful gene editing tool, along with other things. It uses a guide RNA that binds to a Cas protein, directs that Cas protein to a DNA, and then the Cas protein takes over. You can use different Cas proteins to have different effects. Sort of conventional CRISPR-Cas uses Cas9, which cuts both strands of the DNA and leaves the cell to fix things. If you don't provide an alternative, you typically get knockout, where the gene is basically inactivated. If you give it an alternative, you can have something called homology-directed repair that allows for gene editing. However, both of these strategies have issues, and they're not very efficient. More recent strategies use nickase Cas9 proteins, where they basically make a cut to a single strand, and then are associated with an enzyme that makes a modification to the DNA. Base editing, you're changing a single nucleotide to make a point change, typically with a deaminase. With prime editing, this is going to use reverse transcriptase and a provided template to make larger changes. Unlike in the case of your conventional like gene editing through homology-directed repair, however, the template is coming from the guide RNA itself, not from an external piece of DNA. If you want non-permanent changes, you can also use catalytically dead or decast proteins attached to things like transcription factors or chromatin modifiers. That'll allow you to temporarily change expression. If you want to change what sequence is targeted, you change the guide RNA. And if you want to change what happens, you change the Cas protein. In addition to gene editing, you can also do transcriptional regulation. You can target RNA with specific Cases, and you can do things like use CRISPR as a diagnostic tool. There are, however, challenges. These include trying to make edits that are accurate and efficient, targeting every sequence that we want, so overcoming some of those issues with needing to be next to the PAM, delivering the machinery, evading the immune system. And so there's lots of different strategies, and I'll provide you with links to resources that do a great job about all this. There's a lot more coming down the pipeline, and so I encourage you to check out those free papers. None of this could have happened, however, without the original basic research. When we talk about basic research, it's not that we're saying it's simple. It's often very, very complicated, but it's as opposed to applied or translational research, where you're doing the research for a specific aim, a specific end goal, some sort of application of what you learn. Basic research, on the other hand, is kind of you're more trying to discover something than apply something, typically with the hope that what you discover could be applied in some fashion. So basic research is by no means wasteful. It can lead to really important translational applications. And so let's talk about where CRISPR came from. And so we'll go all the way back to where it came from in bacteria. But let's talk first about kind of how it was discovered. So this is kind of funny because this is the new frontier of genome engineering with CRISPR-Cas9. This paper by Jennifer Doudna and Emmanuel Charpentier, who won the Nobel Prize for developing this CRISPR-Cas technology, discovering it, and then developing it as a gene editing tool. They wrote this, they made their kind of fundamental discoveries in 2012, that fundamental paper, and I'll get to that in a minute. But this paper came out in 2014, so you can see it cuts off here. And this was just with that Cas9, and so, that original Cas9, and so much more has come since then. Before CRISPR, people still were interested in genome editing. They knew that if you made double-stranded breaks in cells, the cells could fix things, and it could even stick in new genes. And so that was kind of some of the earliest work. Then they wanted to see, okay, well, how can we make breaks in specific locations? And so there were things called zinc fingers and talons. You might have heard about those. Those were proteins that were nucleases, so they could cut the DNA, but they didn't use nucleic acid, so they didn't like use RNA guides. They just relied on the protein sequence complementing the DNA sequence. And so they were hard to develop, and they much less efficient, all these sorts of problems, but it was an early gene editing strategy. CRISPR kind of came on the scene and everything changed because now you can make changes that are a lot more targeted, a lot more simply. Because rather than having to engineer a whole protein, you just had to change the RNA sequence. Again, we saw how there's a bunch of challenges, but it's still able to do things. Scientists discovered CRISPRs, and so I'll talk more about what CRISPRs are, before they discovered that it was actually this immune system. And then they discovered that you could harness it as a tool for gene editing. And one of the main discoveries that they made was kind of how you can make a single guide RNA that could target specific sequences. And so this 2012 article, as you can see from the over 12,000 citations and 300,000 downloads, that, and it's open access, so anybody could read it. But this is kind of one of their key papers, and we'll go into this figure and kind of the significance of it in a minute. To really appreciate this, however, we need to go back to the very, very beginning, or at least the beginning of the completed system of these bacteria. And so when we talk about CRISPR-Cas, we're often talking about type 2 CRISPR-Cas systems. So different bacteria have different immune systems. The type 2 CRISPR-Cas system, it has these three main stages: adaptation, expression, and interference. So remember, the goal of this system is for the bacteria to be able to recognize and attack invaders that try to invade them again. So when we talk about invaders, these are typically going to be like a virus that infects the bacteria. So say a DNA virus infects a bacteria and they inject their DNA genome into the cell. The bacteria is then going to take a piece of that genome and stick it into this thing called the CRISPR array. Clustered, regularly interspersed, short palindromic repeats. That's what CRISPR stands for, in case you were wondering. It has these repeats, and in between these repeats are these unique sequences. The sequences are coming from these viruses that have attacked them in the past. Those sequences then get used to make guide RNAs, and the guide RNAs bind to the Cas protein, and the Cas protein uses those guide RNAs to find the virus if it tries to infect it again. If it finds it, then it's going to interfere with it, and it's going to cut the DNA. So let's look a little more closely at these CRISPR arrays. So remember, clustered, regularly interspersed, short, palindromic, and repeating. The repeats, they have these generic repeats, and in between the repeats, you have spacers, and the spacers are going to be the sequences that come from the invader, so from, say, that virus. The sequence in the virus, or the sequence in your target, is the protospacer, and that protospacer gets inserted into this CRISPR array. The protospacer, remember, has to be next to a PAM, and so the protospacer adjacent motif. When the bacteria puts this sequence into their cell, they don't put the PAM in, because if they put the PAM in, then they would basically target their own DNA, which would be bad. So bacteria avoids having that PAM sequence in its own DNA so that it doesn't attack itself. And so it puts the protospacer part into the CRISPR array. And it still has these repeats in between the different spacers, and that's going to help with the processing. The processing. So the cell makes that in the bacteria, it makes the whole CRISPR system as a single operon. So it makes it basically all as once. It transcribes this whole thing as once, and then it processes it. So you have the genes for the Cas proteins, and you have the genes for this thing called the tracer RNA, and then the CRISPR array. This CRISPR RNA, this pre-cRNA, so you have this one long RNA that then gets processed. This trcRNA, this transactivating CRISPR RNA, is one part of the guide RNA in these bacteria, and then the other part comes from this cRNA, which has that has part of the repeating sequence and then the unique sequence, that spacer. This is then going to get this like double-stranded RNA that gets cut by this double-stranded RNA cutter called RNase 3, and that gives you this mature cRNA. And that's bound to the Cas protein to give you these active surveillance complexes. When this complex then goes and it searches the sequences that are in it. So if a virus attacks, it goes and it searches, it looks for that PAM sequence. If it finds the PAM sequence that snaps it to action, it unwinds the DNA and then it cuts. So remember that the spacer was the part basically in the guide RNA that's going to match this protospacer in the target DNA, and it's called into action by first binding to that PAM. When I've been depicting the guide RNA, I've been depicting it as a single piece of RNA. Whereas here we saw that there was the tracer RNA and there was the cRNA. One of the big findings from Doudna and Charpentier in 2012 was that you could actually combine these two into a single guide RNA, this sgRNA. So in these bacteria, they have these two parts, the tracer RNA and the cRNA. Tracer RNA is generic, and then the cRNA is partly has that repeat, that generic repeat, and then part of it has that unique spacer portion. The tracer RNA, part of it is going to recognize the Cas protein to allow it to bind to the Cas protein, and then part of it's going to recognize that generic repeat portion of the cRNA. So it's in these two pieces, which isn't ideal for using it as a genome editing tool. But Doudna and Charpentier show that you can actually combine these into a single guide RNA and sgRNA and use that to effectively target targets. And so here they were showing that they basically had these sequences, these test sequences for GFP, and they were able to use this chimeric RNA that they developed that were able to combine these pieces into a single piece and use it to actually go and cut the DNA. And so that's how we typically use it now is with a single guide RNA. Some Cas proteins actually just like naturally use a single piece guide RNA and recognize structural features and things in order to get it to bind to the Cas protein. But in the Streptococcus pyogenes Cas9, which is the main one that's used in the lab, it does use those two pieces naturally, and so we can combine those into one to make things easier. However, there's still a lot of optimization and still a lot of screening that goes into finding a sequence that will be a good target. It's got to be next to a PAM. It's got to avoid off-target activity, so you want it to be really unique for the site that you want, not bind to other sites. It needs to not fold up into structures that you don't want. There's lots of complications. You need to make sure it actually makes changes that you want and that it doesn't make the changes that you don't want. But it is this really powerful tool. And remember, it comes from that basic research into these bacterial immune systems. So, hope this helped you understand CRISPR-Cas, appreciate it more. Think about where it comes from, think about where it is, and think about where it's going. P.S. I got to meet Jennifer Doudna before she won the Nobel Prize, and so I was actually introduced to her by my PI, so basically my boss in grad school, Lemur Joshua Tour, introduced me, and so it was really exciting. And I got a picture, and I'm really glad I got the picture. Kind of wish I got an autograph, but a picture will suffice.